In this chapter, we're going to talk about pathogenicity and host-pathogen interactions. Really quick, we're going to get into the immune system more in a couple of lectures, but just to give you some terminology at first, in the immune system, we have two kind of arms of the immune system, if you will. The antigen-specific arm and the innate arm. So innate immune cells are things like neutrophils, basophils, mast cells, macrophages. They will gobble up anything that they sense as foreign on a broad sense. So they look at things like LPS or peptidoglycan or certain carbohydrates on the surface of most all bacteria. Okay, they're not specific for any one bacteria. On the other hand, the antigen-specific guys, our B cells and T cells, they are very highly specific for one certain kind of pathogen. And when they see that kind of pathogen, they make a large response. And then after that response is over, there will be some B or T cells that persist that remember that response. They're called memory cells. And memory cells will respond faster if there's a second infection. All right. So pathogenicity is what we're really talking about. That's the ability to cause disease. Okay, you've probably heard the term virulence before, or virulent. That is the extent of pathogenicity. How much disease does this bug cause? Right? There are a series of common steps that occur in any kind of infection. Okay, the pathogen has to gain access to the host. It has to adhere to the host. And then it has to penetrate into cells or tissue or evade host defenses like white blood cells or phagocytes. Phagocytes are any kind of white blood cell that will gobble up something in the environment. And then they cause damage to the host. They do things like use host nutrients, cause direct damage. They produce toxins or waste products, or they can cause hypersensitivity reactions like allergies. Okay, so here's our overall mantra of pathogenicity: entry, penetration or evasion of host defense, damage to host cells, and exit. Okay, we're going to take this step by step and talk about each part of it. Entry is first. So our portals of entry for pathogens. The biggest one are mucous membranes. The respiratory tract, the GI tract, the genitourinary tract, the eyes, all of those are considered mucous membranes. The skin, interestingly enough, is just about impenetrable when it is unbroken. So intact skin is one of your best defenses against infection. Some pathogens can enter through sweat glands and hair follicles. Some fungi can grow on the skin and things like that. But for the most part, your skin is impenetrable if it's not broken, which on the other hand, if it is broken, it becomes a very easy portal of entry for microbes. And we refer to that as the parenteral route. Another thing is most microbes will have a very specific preferred portal of entry. So respiratory viruses enter through the respiratory tract, right? Those sorts of things. It's also a little bit of a numbers game. So if you get infected with like 1 or 2 cells of a bacteria, it's probably not going to cause any disease. They're going to die off before they do anything. The more pathogen you encounter, or the more pathogen that get into your body, the more likely you are to get sick from it. When we're talking about how easy it is to acquire a virus or another infection, sometimes we use the term ID50. That's the infectious dose it's the specific dose that will cause infection in half of the test population. When we're talking about things like toxins or poisons, in toxicology, they also use the term LD50, which is the lethal dose for 50% of the population. All right, so Once a pathogen has encountered a host, it has to attach itself to the host, and that's called adherence. In order to attach, there are adhesins or ligands on the surface of a pathogen, and those bind to receptors on host cells. And a lot of times those are pretty specific, so pathogens generally only infect certain kinds of cells. Right? Kind of like how viral spikes only attach to certain cells, right? Adhesins on pathogens only attach to specific receptors on host cells. Adhesins are mostly glycoproteins or lipoproteins. They vary widely between pathogen strains, but they're found on the surface of the cells. Receptors, same thing, a lot of carbohydrates. They vary between cells, but they're on the cell surface, right? And they will bind specifically. Interestingly enough, if you can alter either the adhesin or the receptor, you can prevent adherence. And that's how some antibodies will work. Some antibodies will prevent the ligand from binding to the receptor by either binding to the receptor or binding to the ligand. Here is a list of different pathogens. Their adhesins and their ligands. This is just if you're curious. There's things like E. coli here, Streptococcus, Yersinia pestis, causing the Black Plague, Staph aureus, Salmonella. All right, so we have to bring up biofilms again here. Biofilms are very good at adhering to a surface, right? So the bacteria adhere themselves, they secrete the glycolax, and then more bacteria adhere. It can be several layers thick. It can contain lots of different species of bacteria. They're resistant to things like disinfectants and antibiotics. We know that already. E. coli can also make biofilms in the intestines. That's just one example. All right, avoidance of host defense and penetration. So you could have the pathogen. The pathogen encounters the host. It then adheres to host cells, okay? And then it has to get around the immune system, okay? Or get into cells. And there are different tools that pathogens can use to do that. The first one is the capsule. So if you have lots of carbohydrates and proteins on your cell surface, that could potentially signal to the immune system. One way to get around that signaling happening is to cover up most of that with glycolax. So some cells can secrete that glycolax, and it actually will kind of shelter them from the phagocytic cells of the immune system because it hides the antigens away. Some pathogens have different components of their cell wall to help them avoid host defense. There's a thing called the M protein, the OPA protein, mycolic acid, which we've talked about. Mycolic acid will function kind of like a capsule, but it just helps resist being taken up by a phagocytic cell. Then we have our exoenzymes, so extracellular enzymes, things that are made inside the cell and then secreted, that help it get into tissues and cells. So we've got coagulase, which can clot the fibrinogen into fibrin, right? This helps isolate the bacteria and protect it from the host defense. We have kinase. Kinase can digest fibrin and clots formed by the body, so kind of the opposite of that. Our body will work to try to isolate an infection so that it doesn't spread. So kinase is one enzyme that can try to get around that. IgA protease is another one. So IgA is a kind of antibody found at our mucosal sites. Some pathogens actually secrete enzymes that destroy those antibodies, and that enables the pathogen to better adhere to mucosal surfaces and then cause infection from there. And some bacteria can even make collagenase, which breaks down collagen, right? Collagen forms the connective tissue between our cells. And if you are trying to get through that tissue, breaking down the collagen will make it much easier for you. Another way that bacteria can evade the host defense is by changing up the antigens on the surface of the cells. So essentially over time, you start with one set of proteins and carbohydrates on the surface of your cells, right? The host will make an immune response to that. Most of those will go away, but then you'll get a new population of cells that have slightly different proteins and carbohydrates on their surface. And so that can cause another infection, okay? Kind of like antibiotic resistance, but a little bit different, in that it's the same pathogen. It just looks a little different every time the infection comes up. All right, so how do you get in? Once you attach to the surface of a cell, a lot of times that will trigger a cell to actually send its plasma membrane out and around whatever's attached to it, and then pull it in a vesicle. Let's see. There's a really great video here that I'm going to put in the Canvas module on salmonella. I really think you should watch that. It really does a great job of showing you exactly how salmonella gets in. There's great animations in there. I really encourage you to watch that. Essentially, it shows you exactly how the cell rearranges its cytoskeleton and sends its plasma membrane out around the salmonella and takes it in. All right, so you're a pathogen. You are either inside a host cell or inside host tissue. And now you get to do your damage. Okay. Sometimes that damage is indirect. That pathogen could use the host's nutrients. And in using those nutrients, the host doesn't get them anymore, right? So iron is a big one. We don't have a very large amount of iron in our cells. What we have, we need, though, so it's very important. But pathogens also need iron. So some pathogens are able to essentially take that iron away from our cells by binding to it more tightly than our cells do. Those are called cytophores. So they essentially sequester iron away from the host cell for the pathogen's use. But for the most part, we're talking about direct damage when we're talking about damage by pathogens. So pathogens, like some bacteria and most viruses, will multiply inside cells, right? A lot of times that will make the cells rupture, right? So you get cell death. You get a lot of cell death in a large area, and then you get symptoms, right? Sometimes they can disrupt host cell function. So essentially that means that they can either, in the case of a virus, hijack the host cell's replication machinery to replicate more of itself. They can cause the host cells to kind of tolerate their presence more by dampening some responses that the host cells can make. They can produce waste products or toxins that are harmful. Let's talk about toxins. So toxins are any poisonous substance that can contribute to pathogenicity. So it's a very wide term, okay? Toxigenicity, the ability to produce toxin, toxemia, the presence of toxin in blood. So toxins can be almost anything. Enzymes sometimes, proteins, carbohydrates. What they do is it can be a wide range of things as well. So a lot of times the presence of toxins will cause things like fevers, heart disturbances, diarrhea, shock can also happen if there's a lot of toxins present. Shock is a life-threatening decrease in blood pressure. Sometimes that happens from the presence of bacteria, and that's called septic shock. Toxins can also inhibit protein synthesis, so potentially stopping the production of enzymes that could break down that pathogen or that toxin. Some toxins will destroy blood cells, either white or red. Some toxins can disrupt the nervous system, and some can damage cell membranes. And we know that if cell membranes are damaged, cells are more likely to die. There are two big classes of toxins, exotoxins and endotoxins. As the prefixes apply, exo are secreted out of the cells and endo are part of the cells. Okay. So exotoxins are things like enzymes that are made inside a cell and secreted out. Endotoxin specifically is lipopolysaccharide. Endotoxin, remember, lives in the outer membrane of gram-negative bacteria, right? Outside of that small layer of peptidoglycan, there's that outer phospholipid bilayer that has lipopolysaccharide, or LPS, inside of it. When that cell is intact, the LPS is harmless, but when that cell starts to die and little pieces of it break off, that's when it becomes a toxin. We'll talk more about that in a minute. We'll talk about exotoxins first. So most of the time, these are enzymes secreted by bacteria into their surroundings, right? They change the metabolic functions of cells in the environment, right? Sometimes the genes for toxin production can be on plasmids, and they can be transferred from one cell to another. Gram-negative and gram-positive bacteria can make toxins. We can make antitoxins, either in our bodies or in a lab, which are essentially antibodies that bind to toxins to prevent them from acting on other places in the body. If you're making that an inactivated form of the toxin and you're using that to prime the immune system against a potential pathogen, that's called a toxoid, and that would be used in something like a vaccine. There are 3 groups of exotoxins. For the most part, the names of the groups tell you what they do. There are A/B toxins, membrane disrupting toxins, and superantigens. Most exotoxins, I should say, are A/B toxins. And that essentially just means there's 2 parts, an active enzyme component and a binding component. So you have to bind to the host cell and enter, and then you alter host cell function with your enzyme. Membrane disruptors, just like it sounds. They can kill phagocytes or white blood cells that are trying to take them up to destroy them. They can cause protein channels to happen in a membrane and like poke little holes in the membrane to try to kill the cell. Anytime you disrupt a cell membrane, you kill the cell. Then we have superantigens. Superantigens cause a very robust immune response. So robust that it's no longer a good thing. Okay, too much immune response is bad. So a normal amount of immune response will kind of alert the immune system that there's a pathogen present, and it will work to get rid of that pathogen and clear the infection. When the immune system is really overstimulated, it causes a huge release of pro-inflammatory proteins called cytokines, and they can cause a lot of very bad symptoms that come with an overactivated immune system. So fever, nausea, vomiting, diarrhea, those will happen in any infection, but they can be worse with a superantigen because the immune system is really ramped up. It can even cause shock and death. One group of bacteria that make superantigens are the C. diff bacteria. So Clostridium difficile, you guys know it's an intestinal bacteria. It's one of the bad bacteria that can overgrow and the good bacteria are gone, right? So it's not just the presence of the bacteria that is harmful, it's actually the toxins that C. diff makes that cause all of that inflammation in the gut. And the toxins can persist even after the cells have died. So that's another thing to keep in mind too. So while a patient may have cleared the infection, there might be toxin left behind that's still making that inflammation happen. And sometimes they actually give antitoxin to try to get rid of that. All right, endotoxin. Endotoxin is lipopolysaccharide, right? Here is our gram-negative cell wall and outer membrane, right? So there's a peptidoglycan, here's the outer membrane, and you have all of these little pieces of lipopolysaccharide in it. When the bacteria is intact, all of that lipopolysaccharide stays put, and it doesn't trigger a huge immune response. When that bacteria is dividing, or when that bacteria has died, little pieces of LPS will free themselves and be floating around in the environment. And that is a huge trigger to the immune system that there's a pathogen present, because it's only found on gram-negative bacteria, and it really ramps up the immune response. All right, summary slide on exotoxin, a little bit more about endotoxins. Again, they can be a super antigen. They can cause macrophages, which are innate immune cells, to release a high level of those cytokines, those pro-inflammatory proteins. They can activate blood clotting. It can produce things like endotoxic shock. It can even weaken the blood-brain barrier. Okay, and if there's enough present, it can cause death. Something interesting about endotoxin is that it's the glycoprotein, right? It can be present in sterilized materials because it's not part of a living cell, right? It can be free floating. And so it can be present when no other bacteria is, and it can still cause harm in that state. Really quick about fever. Endotoxin that is released can be taken in and digested by white blood cells of your immune system, the phagocytes. And those phagocytes can then make cytokines, pro-inflammatory proteins, that travel to the brain and cause the hypothalamus to increase temperature. That's a long story short by a long shot, but that's kind of the overall what happens. And cytokines, I'll point out, is just kind of an umbrella term for lots of different kinds of pro-inflammatory proteins. Some of them will activate T cells, some of them will activate B cells, some of them will activate innate guys, and some of them, the ones that we're talking about here, will actually travel to the brain and raise the temperature. Okay, there's a video here on endotoxin that I'll also link in the Canvas module. All right, so some toxins wouldn't be possible without plasmids, right? We know what plasmids are already. We know that they can carry genes for extra things, right? Like toxins or antibiotic resistance. So I'm going to skip over most of this, okay? Viruses are a pathogen, right? We already talked about viruses recently as well. And so we know that they attach to their host by spikes, right, on the surface of the virus. They go into the cell, they replicate, and a lot of times they'll cause cell death. And that cell death can result in what we call cytopathic effects, or visible effects of viral infection if you're looking at cells and tissue. You'll see areas of cell death. You can see a change in cell function. Sometimes we'll actually see oncogenesis happen. The formation of kind of cancerous cells that have lost their contact inhibition, meaning that they just keep growing and they don't stop. That's how tumors start. Sometimes we'll see the production of proteins called interferons. Interferons are made naturally by cells that are infected with virus to try to prevent their neighboring cells from getting infected. It can also cause changes in proteins present on the surface of the cell, and sometimes that can alert the immune system that there's a virus inside that cell. All right, fungi can be toxic as well. Not only do they grow in and on tissue, but they can make toxins as well. For example, ergot toxin grows in rye. There was a large outbreak of ergot toxin on rye grain in the Middle Ages. It's actually the natural source for LSD. It causes hallucinations. Aflatoxin is a very carcinogenic toxin on plants like peanuts. Mycotoxins are produced by mushrooms, so specifically the mushrooms that will probably kill you when you eat them. There's lots of different toxins that mushrooms can produce. Algae can make toxins as well, neurotoxins and the toxins that cause red tide. Protists, while they are kind of their own unique thing, they can invade host cells. A lot of them will reproduce inside host cells, Some of them can even reproduce in your white blood cells. They can hijack your white blood cells and reproduce in them. Sometimes their waste products can cause symptoms and be toxic, and they can make their own toxins as well. All right, helminths are hard to get rid of, right, because they are eukaryotes. They use host tissue and host nutrients. Their waste can cause symptoms, and a lot of times just their presence, the mass of the worms, can cause symptoms as well. All right, so the last part is exit. So pathogens have a preferred portal of entry. A lot of times they're going to use that same portal to exit. Okay, so respiratory pathogens, right? They enter by being breathed in. They exit by being breathed out, sneezed out, coughed out, in droplets, right? Gastrointestinal pathogens, they usually are spread through the fecal-oral route. Now, the fecal-oral route is a lot less direct than it may seem by the words. So anytime someone has a bit of fecal matter or fecal bacteria or something on their hands, maybe they didn't completely wash their hands, right? They touch a surface, that bacteria gets deposited on that surface. You can then touch that surface, pick it up, and if you don't wash your hands before you eat or things like that, you can put that bacteria into your body. So it can be a very indirect spread, but that's how most GI bugs are spread. Genitourinary, just as it sounds, a lot of STDs, right? Mucosa to mucosa, right? Secretions to mucosa, that sort of thing. Blood is another big one. Use needles or syringes, but also mosquitoes. Mosquitoes are essentially dirty needles flying around, right? Or drainage from wounds. So here's that overview again. Entry, penetration or evasion of host defense, damage, and exit. So understanding these parts will help us understand how our body can react. And then we'll talk about the immune response in a few lectures. Okay, thank you.